Hello and welcome to Chapter 5. So today we're going to start that, and I think this is probably just going to be 2 lectures. We'll see how it goes, but I can't imagine it's going to be longer than 2 lectures. But let's talk about what it is that we are going to talk about. So as I said, this is Chapter 5. It's the integumentary system, and we'll be dealing with the lecture objective set questions for that chapter. And you can see here they're all listed out. It's not a particularly long list. I don't deal with a lot of the material that's in this chapter. I just focus on some of the things. So we'll take a look at that and we'll begin right now. So let's do that. And let's go ahead and label our notes properly. So what we're going to label them is this is the integumentary system. Otherwise known as the skin. Okay. So let's take a look first of all at an image, and we're going to be looking at this image a number of different times, but let's begin with looking at it first, and then we'll get some information in our notes, and we'll come back and look at it again. So this is the image that's in your textbook, and it is the image of the skin as well as the subcutaneous layer. So by now, hopefully, you're familiar with that term, subcutaneous layer, because we've run into it several times when we talked about location, for example, of adipose tissue. So let's break down this drawing and see what we have here. So the skin proper is composed of 2 layers. This layer, which is the epidermis. Epi mean above, and what it's above is the dermis. So above the dermis, and that's this layer here. This is all epithelial tissue. And we'll describe the structure of these epithelial cells. This is stratified squamous epithelium. And then below that is the dermis. So epidermis and then dermis. And notice how thick that dermis is. So this is a structure that's quite a bit thicker than the epidermis. Both of these are going to vary in thickness depending on where you are in the body. When we look at the dermis, however, we can divide that into 2 regions, the so-called papillary region and the reticular region. So each of those are part of the dermis. The papillary region is this area where you see these hills and valleys and hills and valleys that you see here. That region, if we draw a line along sort of roughly around the bottom of the deepest inward projection of the epidermis, that would roughly be where the papillary region is. So that is the region of the connection between the epidermis and the papillary region. And then the other part of it, most of it, is the reticular region. So what is going on here with this rough, sinuous connection between the epidermis and the dermis? And the answer is really quite simple. It's a way in which we can glue this layer of epithelial tissues much more securely to the underlying dermis. So we're creating surface area here. We're creating more surface area here as we undulate that surface rather than having a simple flat surface. Okay? So this is all stratified squamous epithelium and has structure. And we'll look at that. But within the dermis, we get more structure in the sense that we have more things going on. For example, you'll notice that the hair and the structures associated with the hair are all found in the dermis. We also have some glands, so the apocrine glands, eccrine glands, and all the other structures that are associated with the blood vessels and so forth and so on. If you look carefully right here, this is a fairly complex capillary bed here close to the surface, and that's there to provide nutrients and oxygen and take away waste for the epithelial layer, because as we've learned, the epithelial layer will not have will not be vascular, so there won't be blood vessels within this region, and so diffusion is going to be the primary structure or the primary way in which these cells find the necessary oxygen and nutrients they need to survive and to get rid of waste. So that capillary bed is there for that reason. Further down, there's another capillary bed here. And this is more or less embedded in this layer, which is deep to the skin. So as I mentioned, the skin includes the epidermis and the dermis. And then below that is the subcutaneous layer. And the subcutaneous layer is, strictly speaking, not part of the skin. It's not considered to be part of skin. We will talk about it fairly briefly here because it's so closely associated with the skin that we can touch on it right now at this point in our discussion of the skin, or not in this chapter, I should say. We'll talk about that subcutaneous layer, which is deep to the skin. So we've got a lot going on, and if you notice that there is a pretty extensive capillary bed in this region as well. So one of the things that happens is that this capillary bed, being close to the surface, is a way we can dump heat. So when we're hot, as you might know, is we tend to be flush. That is to say, we tend to have a rosier, brighter, pinkish color. And that's because there's more blood in this capillary bed. The body is transferring heat closer to the surface so that it radiates away and we can get rid of some of that excess heat. Now, when we're cold, we tend to be paler and more ghost-like in our appearance. And that is because these capillary beds tend to shut down a bit, and blood tends to be more concentrated down here in the deeper regions, subcutaneous layer, and that is to conserve heat. So that's one of the things that your body can do, is they can redirect blood supply. During times when you need to get rid of a lot of heat, there's more blood circulating through the capillary beds here close to the surface. When you're trying to conserve heat, that blood tends to be conserved more down here in the lower capillary beds. It's not that there's no blood up here, because we do have to maintain the living tissue here, but we can control the total volume of blood, and we can make this more or less in terms of volume for blood. So we'll talk about that. So that's the basic structure. By the way, in this corner you can see that intersection between the dermis and the epidermis, and these kind of nipple-like structures that interlock, and those are called the dermal papillae. All right, so let's get some information down into our notes, and we'll start off with the anatomy of the skin. The anatomy of the skin. Okay, so kind of an overview first. So let's remind ourselves what an organ is. And there's a reason we want to do this at this point. We probably know what an organ is, but let's just remind ourselves what the definition of an organ. So two or more tissue types joined together for a specific function. So that's kind of a shorthand. I mean, we can get a more elegant definition, but that's enough for us right now because we're just kind of reminding ourselves of what an organ is. And why did I ask that question and why are we there? Because we're going to make this statement. The skin is the largest organ of the body. The skin is the largest organ of the body. So how can we make that statement? We're calling it an organ. Well, it fits this definition here. So quickly, let's go back and look at that drawing that we were just looking at and see if that fits in with what we're seeing in this drawing. So different types of tissues. Well, first of all, we have epithelial tissue here. And then we have connective tissue. So that's it right there. We've met that definition, but there's more to it. Because notice this. This is a smooth muscle. You can see over here. It's the rector pili muscle. And it's connected to the base of the hair. We also have nerves here. Let's see if they call them out here. Yep, there it is. So there's a corpuscle of touch, or Meissner corpuscle. That's part of the nervous system. So you can see we have all of the four basic types of tissues. We have epithelial tissue, we have connective tissue, we have muscle tissue, and we have nerves. Okay? So it fits the definition. This is an organ. All right, moving on. So, largest organ system in the body. And its surface area is, on the average, around 2 square meters of surface area. That's about 22 square feet. It's a pretty large surface area. Right? It weighs in around 4.5 to 5 kilograms, which is around 10 to 11 pounds. Okay. Its thickness is going to vary depending on where you are in the body. So anywhere from 0.5 to around 4 millimeters in thickness. Okay. Depends on location. It is composed of 2 parts. One is the epidermis. And the epidermis is the superficial layer and it is epithelial tissue. The second part is the dermis. And the dermis is connective tissue. And some of its fibers are going to extend down into the subcutaneous layer. And thereby anchor the dermis to the underlying tissue, which we're calling the subcutaneous layer. Now that's the skin. These two things make up the integumentary system. I'm going to add, though, a third thing on our list, but I want to emphasize this is really technically not part of the skin, but we are going to mention it and talk a little bit about it. And this is the subcutaneous area. This will be deep to the dermis. Okay. It is also called the hypodermis or the superficial fascia. So it has three names for the same thing. Why are there three names for the same thing? Well, that's the way things developed over time. Both of these terms, the hypodermis and the subcutaneous layer, are terms that are referencing the skin because they obviously, in this case, they actually mention the skin, the dermis, and this cutaneous also refers to the upper layer of the skin. So below the skin, below the dermis. Superficial fascia, on the other hand, is referencing the underlying muscle tissue. So people who study muscles, who describe muscles, who work with muscles, they would not call this layer the subcutaneous layer. They would call it the superficial layer because their frame of reference is the muscles that they're studying. This terminology is often used when we're thinking about the skin or the relationship of this structure to the skin. So, but basically it's the same thing regardless of how you call it. It's the layer deep to the skin. Okay? This layer, the subcutaneous layer, areolar tissue, consists of, number one, areolar connective tissue. Adipose tissue. And sometimes the deeper hair, the bulb of deeper hairs are actually in the subcutaneous layer too. So it is a structure that is closely associated with the skin. Okay, let's talk a little bit about functions. We're just going to list them here because later on in the lecture we'll get into more details of the functions, but we're going to simply list them right now. Number one, we've got body, temperature, regulation. Number two, we have it's acting as a reservoir for blood, and we're going to expand on that idea later on, as I say, when we get into the details of the functions. Number three, protection from the external environment. Number four, cutaneous sensations. We can feel things through the skin, and it gives us lots of very important information about our surroundings. Number five, excretion and absorption. I'm going to put parentheses around here. In fact, I'm going to put a little question mark at the end and put a parenthesis around that. And I'll come back to why I did that in just a second. Let's get the sixth one down. The sixth one is that it's involved in vitamin D synthesis. Okay. So when we get into the details of functions, I'm going to come back and discuss this, why I have kind of questioned that. Your textbook mentions it, talks about it, so I put it down there. So I'll just leave that hanging there for a while, sort of whet your appetite for that mystery. Okay, so these are the functions. Let's take a look at the epidermis then. I'm going to talk about the epidermis. The epidermis. Let's come back to our picture and remind ourselves exactly what we're talking about. So it's this upper layer here. We're going to get into details now. We're going to get into the weeds and talk about this layer. We're going to break it down. We're going to look at the cells it's composed of and the arrangement of those cells and how it all functions. And then we'll focus on the dermis. And then finally, we'll talk about the hair and the glands that make up the skin. All right, so when we look at the epidermis, we find that it is composed of 4 types of cells. 4 types of cells. And let's go ahead and I'll quickly show you each of those types. And again, I'm going to come back and we're going to look at this again after we've talked about cells to remind ourselves of what they look like. So we're going to talk about this cell, which makes up most of the cells of the epidermis. This is keratinocyte. And we already are familiar with this term, the intermediate filaments. Remember, that's part of the cytoskeleton. So this is a keratinocyte, and that makes up about, somewhere around 90% or so, maybe 80% of the cells of the epidermal layer. And then we have something called a melanocyte. And these little extensions that you see here and little granules that you see there, those are all important. So we'll talk about that, what this means. So this is my melanocyte. And then we have something called a Langerhans cell. And we'll talk about the significance of this cell. It's part of the immune system. We'll look at that. And then we have a Merkel cell, which is the one in purple. And then there's the tactile disc, which is part of a sensory neuron. So this is part of the sensation of touch. And these two interact to give you that sensation of touch. So we'll take a look at that. So those are the 4 basic types of cells. Keratinocyte, melanocyte, the Langerhans cell, and the Merkel cell. And we'll, as I say, come back and look at those again in just a moment. Okay, so let's go through and describe a little bit about each of one of those types of cells. So we'll start off with the keratinocyte. Okay, these make up around 90% of all cells in the epidermis. They produce a protein, and we'll talk about the developmental pathway of that protein in just a bit. And that protein is keratin. As you know, the skin is keratinized stratified squamous epithelium, and so these cells, these keratinocytes, need to produce the keratin. In fact, that's what they're named after. And keratin is a tough, fibrous protein. Okay? And it protects, it has a protective functioning in the skin against abrasions. And what is an abrasion? An abrasion is a type of injury in which you scrape yourself, as opposed to something like a cut. So if you fall down and skin your knee, that's an abrasion, as opposed to accidentally cutting yourself with a knife. That would be that would not be an abrasion. So an abrasion, heat, microbes, and chemicals. So it's kind of an all-purpose protein protecting us from many different types of injuries or problems that the skin might run into. So abrasions, heat, microbes, and chemicals. That's keratin. Also, they produce another substance, and this is the lamellar granules. And this is a lipid. No, this is a lipid. And it is a water resistant or repellent, as your book mentions, sealant. So the laminar granules contain a lipid substance that is acting as a water repellent and sealant. And what it does, its purpose, so what happens is it is through exocytosis. It's released and fills up the space between cells. And what it does is it decreases water loss or gain. So it helps to make the skin watertight. So both in terms of gaining water and losing water. So decrease water loss or gain. And it inhibits the entry of foreign substances. You know, because it is a sealant. The keratinocytes are anchored together by desmosomes. So we've talked about these. You know what they are. And we'll actually see the evidence of those desmosomes when we look at the structure of a prepared slide of the skin. All right. The second type of cell is the melanocytes. And this makes up about 8% of all cells in the epidermis. And these cells produce a pigment. Oddly enough, this pigment is called melanin, and that's the name of the cell. They have long, slender projections of the cell membrane. And what they do is they transfer this pigment to keratinocytes. And approximately, for every melanocyte, we have approximately 36 keratinocytes. Per melanocyte. So each melanocyte is going to service approximately 36 different keratinocytes. Okay. So when we look at this cell sitting by itself, it has these long, slender projections of the cell membrane like this. Okay. Here's my basement membrane. And then we get the keratinocytes. That are in and surrounding these projections, not in, but surrounding the projections. And what's going to happen is we're going to transfer. So if the orange is the pigment, melanin, that is going to be transferred then to all of these cells through these long projections. And then what's going to happen, so we get this movement of this material into cells. And then if we look closely at one of these melanocytes, one of the keratinocytes, and here's my nucleus. The keratinocyte is going to create an umbrella of this pigment over the nucleus. And the idea is that ultraviolet radiation is going to be prevented from damaging the nucleus with this umbrella of pigment sitting above the nucleus. So, let's get that information into our notes. Melanin is a yellow or brown black pigment that can contribute to skin color. And absorbs ultraviolet radiation or light and protects DNA from damage. Because ultraviolet radiation is a mutagen. It can literally change and cause mutations in the DNA. And to prevent that, we are going to have this protective umbrella of this pigment over the DNA, over the nucleus. The third type of cell is referred to as the intra-epidermal macrophage, also known as the Langerhans cell. Same thing. This is the older term. This is the newer term. And what's been happening in the science of naming things in the human body is that there are many, many structures in the human body which had names of the scientists who identified and described them in the literature, so they added their name. So somebody with the name of Langerhans actually was able to identify these cells, named it after themselves. I don't know if it was a man or a woman, probably a man, because it was dominated by men early on anyway. And so add their name to this. But now they're going back and changing that. So they're changing the names of structures to names that have more meaning. Intra, between the epidermal cells. A macrophage that's in between the cells of the epidermis. And what they're talking about is in between the keratinocytes in particular. Anyway, these cells actually arise from bone marrow. So they're actually manufactured in bone marrow. And then they migrate to the epidermis. And there they can intercept antigens. So they can trap antigens. that penetrate the skin. Okay. And then they transport the antigen to local lymph nodes. And then they will interact with helper T cells. Now, this is a very complex story, and you're going to learn about that story when you do the immune system. But I'm going to give you a very short sort of synopsis of what we're doing here. So this is a macrophage. The macrophage will engulf things. So it's going to leave, it's going to arise in the bone marrow, travel through the blood, get to the skin. It lives in the skin, it lives in the epidermis until at such time, for whatever reason, you've damaged the skin, you've punctured the skin, somehow some foreign antigen, which is a foreign substance not supposed to be in your body, gets into the skin, and then the macrophage will engulf that material. And once it's been engulfed, then the macrophage will migrate out of the skin and go to a lymph node. And in the lymph node, it will find some helper T cell that will actually recognize the antigen. And then you will have immune response. So it's part of the immunity of your body. A very simple way to think about this is if, and I usually do this in a face-to-face class, and I pick on some student, and I say, okay, this student here wearing the baseball cap and having a blue jacket on and the tennis shoes, say that person is a foreign microbe that has penetrated the skin, and I'm a macrophage. And a macrophage will go in and I will eat that student, and I will digest that student, except that with the hat and the jacket and the tennis shoes, I will take those and I will hold them out and I'll display them on the surface of my skin, of the cell membrane. I will literally hold those things out. Then I will leave the classroom and I will go to another place in the school in which there is some individual that says, oh yeah, I recognize that hat, and I will interact with that person, and then that person, the helper T cell, will become activated, and it will do a number of things to begin the process of defending yourself against something that has that hat or has that tennis shoe or has that blue coat. Okay, so that's in short what's happening here. These are the intraepidermal macrophages, and they're part of the immune system. And it makes sense, doesn't it? Your skin is this barrier, but if it's broken, things can get through that barrier, and then you can intercept those things and start some sort of response to protect yourself from disease. All right, so that's what we're talking about there. So the fourth and final cell is the tactile epithelial cell, also called the Merkel cell. Okay. This is located in the deepest layer of the epidermis. And it forms an association with a nerve, and it's going to interact with that nerve and what is called the Merkel disc. And this is actually part of the nerve. Okay. And this produces the sensation of touch. So basically what you have is a cell, and that is my Merkel cell. And then you have a nerve that has this like disc-like structure. And that right there is the Merkel disc. And it's this interaction between these two things that gives you the sensation of touch. Okay. All right, let's go back and look at those pictures now that we've had a description of each of the cells and see what we can see now as we look at this picture and bring in some of the ideas that we've discussed in terms of how these cells look and how they behave with looking at the images of the actual cells. So here we are. So this is the keratinocyte. We don't have a lot of information here. I do want to tell you that this image is probably not the best image because a typical keratinocyte would not have these spiky structures. It would be filled out. It would be a nice looking cell. It would be very flush and big. This is shrunken, and when it shrinks, the tonal elements in here, the intermediate filaments, which is going to be keratin, causes these spiky things. So as the membrane shrinks, these hard fibrous proteins that are in the cell do not shrink, and therefore it's like a tent here with the center pole of the tent holding up the top of the tent. Here is my melanocyte. There you see the melanin granules. There would be these cells here, and these things, these processes would be transferring that pigment into these cells to protect the nucleus. Okay. Here is that Langerhans cell. This is the macrophage that we were talking about. It's part of the immune system. It'll engulf things and then display different antigens on the cell surface, leave the skin, and go activate the immune system. Here is my Merkel cell, and there is the sensory neuron with the tactile disc. Okay. So that gives you a good sense now of the kinds of cells we're going to look into, or we're going to see once we get into the epidermal layer. So now we are going to describe the epidermal layer. And before we get into writing notes, let's go ahead and take a look at an image, a picture, if you will. And here, side by side, is the actual image of a section of the skin. Here is the basal surface. There's my dermal papillae. So this is the dermis extending upwards and creating these ridges and these interlocking devices that you see here. And then if we turn to this image, which is the drawing of that, we can begin to make out some of the cells we've just talked about. So let's go ahead and do that. So here is my melanocyte. There is the processes. You can see that it's extending itself into this layer, transferring the pigment into these cells. Here is the sensory neuron, the Merkel disc, and the Merkel cell. As I mentioned, it's in the deepest layer, which is this bottom layer right here. So this would be my basement membrane here. There's the Langerhans cell. And all the rest of what you're seeing here are keratinocytes. Now, if you look closely, we can see very distinct layers, and that's what we're going to talk about. So we're starting from the top. We have something called the stratum corneum, then the stratum lucidum, then stratum granulosum, stratum spinosum, and then the stratum basale. So those are the layers that we are going to describe now in our notes. Each of these layers, there's something different happening. Each of these layers has a part to play in the development of this layer. One thing that you'll notice is that by the time we get to the top layer here, these are dead cells. These are dead keratinocytes. And this is where the cells become very flat and very numerous, stacked on top of each other. And you can see that there are not interlocking, but they're overlapping one another, as you see in this drawing. And so this is the surface of the skin. And as we go deeper, these are living cells. These are also living cells. And then as we progress further and further away from the source of blood, the cells begin to die, and they go through a developmental pathway, which we'll be describing. Now here, I do want to point out something in this drawing. So if you look very carefully, you'll see that there are spaces between the cells, and then they've drawn in these little lines that you see there. And what they're trying to represent there is this image right here. So, as I mentioned, a normal keratinocyte would not have these little spiky structures here. It would be a nice round cell, but when we take this material, when we take skin, and remove it and prepare it for observation using staining techniques and what have you, what happens in this one layer here is that the cell membrane shrinks. But these cells are connected by desmosomes. And so where these two neighboring cells are connected, we have this very strong connection which stays in place. And so basically what's happening is the cell membrane is shrinking away from the connection between the cells, the desmosomes, but not breaking that connection. So we end up with this spiky appearance that you see right here. And we'll address that. So let me very quickly kind of draw a nice picture to try to illustrate what we're talking about. So if I have two cells, and they're nicely formed, like this, and we'll just, for the sake of argument, I'll say I have two desmosomes right there. And then when I remove the tissue, I prepare it, put it on the slide, stain it, and so forth and so on, these cells, because of the state that they're in, the developmental pathway that they're in, the membrane is literally going to shrink, and we're going to get this kind of spiky appearance between the cells. And so these two things represented right here, so that's my desmosome. And the cell has shrunk away, but not entirely pulled away. Okay. And it leaves that little spiky appearance. This is only found in the prepared dead tissue. In a normal cell, or normal tissue that is still living, you would not see this structure. So it's called an artifact, as I've described before. But it is diagnostic. It tells us something about what's happening in this particular layer, which happens to be the stratum spinosum, and spine, spinosum. It's named after that spiky appearance, as you can see there. Okay. All right, let's go through then and talk about the epidermal layer and fill in some of the information. All right, so come back. You already have this in your notes. I'm repeating myself here. So this is going to be the epidermal layer. And I've got a couple of observations that we need to keep in mind. Thin skin and thick skin refers to the thickness of the epidermis. So when I say thick skin, thin skin, we're referring to the thickness or lack thereof of just the epidermis. However, thickness of the skin is going to vary. And that variation in thickness is related to both the thickness of the epidermis and the thickness of the dermis. So both things can vary. But when I talk about thin skin or thick skin, it's what we're talking about is the epidermis. Okay? So let me give you some examples here. So between, for example, the scapula, the dermis is especially thick. Whereas in the palms and the soles of feet, the epidermis is thick. So variations in thickness in terms of the entire structure that we call the skin. And each varies in thickness depending on where you are in the body. But the actual terminology, thick skin, thin skin, is in reference to the epidermis only. All right. So now we're going to get into a discussion of the layers. So the epidermis is composed of 4 to 5 layers of cells. 4 layers in most of the body. But 5 layers in thick skin, such as the palms and the soles. Okay. So we're going to now survey these layers. So we're going to do a survey of the 5 layers. We're going to do all layers. And we're going to go from deep, the deepest layer, to superficial. We're going to start from the bottom and work our way up. From deep to superficial. And let's once again return to the image. And so what I'm going to do in our notes is I'm going to start here. This is one of the layers, stratum basal. We'll talk about that. Then we'll move on and talk about the next layer, stratum spinosum. And then the next layer, stratum granulosum. And then the stratum lucidum, this is the layer that may or may not be present. So in places like the palms and the soles, we will have a 5th layer, but most of the skin only has 4, which would be the stratum corneum, stratum granulosum, stratum spinosum, and stratum basal. Okay, let's begin. And I'm going to go ahead and write out description of each of these layers, and you should have this, and if you don't, you should probably stop now and get this image, because we're going to go through and use this as our to fill in some information, so you'll need that. It's part of the handout, the lecture handout in this section. So make sure you have that available. We're going to need it in just a moment. So let's begin. Number 1, it is the stratum basal. This is a 1 layer of cuboidal shaped cells. So actually it's cuboidal, from cuboidal to columnar. It varies, but between those shapes. So sometimes more elongated, sometimes less elongated. Okay. This layer is mitotically active. And it can be thought of as the stem cells for the upper layers. Of keratinocytes, I should add. Also within this layer, we are going to find a couple of other cells. Number one, we're going to find some melanocytes. And we're also going to find some tactile cells. In this layer, the keratinocytes contain scattered intermediate filaments, which are called tonal filaments. All right. So, at this point, since we've described this layer, let's go ahead and I'm going to color this layer in. Here it is. We're going to identify that layer. And then I'm going to identify within that layer a couple of different cells. There's one there, and there's one right there. Okay. And so let's go ahead and identify what those are. Okay. So this is the stratum basale. And we have the melanocyte. And we have the tactile cell. And associated with that tactic cell, as you can see, is our nerve. And here I want to point out something. I'm going to do this in blue so we can see it. We have cell division. Okay, so the production of new cells. So, and this happens in a lot of instances. The bottom layer of a tissue is often the layer that is mitotically active. So there is some cell division, as we'll see up in this layer, but most of it is going to take place in this layer. And when cell division takes place, mitosis, you're going to produce two daughter cells. One of those cells is going to remain in contact with the basement membrane. The other one is going to be part of the column of cells. And as that cell moves forward, it's going to be further and further away from the source of nutrients. And as a result, inevitably, as that distance increases, these cells are going to die simply because they don't have enough nutrients to maintain metabolism. And so it's the cell that maintains contact with the cell membrane that becomes or becomes the stem cell, that becomes the source of new cells. And so if you recall our discussion in the last chapter, the basement membrane plays a very important role in the development and growth of tissue. And this is one of those ways where the contact with the basement membrane is important for this cell to maintain its ability to divide and provide new cells for the cells above it. Okay, so that's that first layer. Okay. All right. The next layer, layer number two, is the stratum spinosum. And this is a layer of 8 to 10 cells. Okay. There is, as I mentioned, some cell division. It's reduced. But it's reduced. Okay. The shape of the cell is polyhedral. And in slide preps, the membrane of the cell shrinks and exposes the connection, or exposes the desmosome connections between cells. Okay. And these spiky areas are, within them are these bundles of intermediate filaments. That produce spikes. Okay. All right. Furthermore, the membrane processes of melanocytes project into this layer and we get the transfer of melanin to the keratinocytes. All right, so let's take a look. What layer are we talking about? The stratum spinosum. And we can see that spiky appearance. Okay, that's this whole layer here. And we can also see, if we can zoom in enough, we can now see that this melanocyte, the processes are extending into this layer, and we're transferring that pigment to the keratinocytes there, as you can see. What else can we say? Well, let's go ahead and bring in our drawing. So take a look at our drawing here. All right. And what color are we going to do this? Just do this in green. So here, I'm not going to try to do the whole layer, but basically all of this layer right here. That layer there. And what are we going to call that layer? That's going to be a stratum spinosum. have the extensions of melanocyte there. And also, you may notice we have our Langerhans cell. Well, there we are. All right, so that's the second layer up. Let's move on. We talk about the very next layer, which would be number 3. And this is the stratum granulosum. And this is a layer of 3 to 5 cells. Okay. These cells are now becoming flattened. Okay. And they're undergoing apoptosis, that is cell death. Apoptosis. And that cell death, as they move further away from the source of nutrients in the basement membrane. Okay. The nuclei begin to break down, as well as the organelles. That's part of cell death. And we begin to see a darkly stained granules are present. And this is something called keratinohyaline. Okay. And this is a protein. So keratinohyaline is a protein. And what this protein does is it organizes the intermediate filaments into keratin. So we are going to change the nature of those intermediate filaments, and we will call that change, or the result of that change, keratin. Okay. Also present are these laminar granules that we talked about. And these are going to fuse to the cell membrane. And release a lipid-rich secretion. And this is going to fill the space between cells in this layer. And in the stratum, the next layer up, lucidum. And we're going to form a water repellent sealant. Okay. This layer, the stratum spinosum, is the boundary between metabolically active cells, that is living cells, and the dead cell layers in the upper layers of the epidermis. Well, this marks a very important boundary. So the cell layers below that are filled with cells that are still metabolically active. They are beginning to die in this layer, in the stratum. I'm sorry, this is not stratum spinosum. This is the stratum granulosum. Let's see, I want to make sure I did that right. No? Yeah. Did I mismark that? Let's see. Yeah. So I screwed up here, didn't I? 3. No, I don't think I did. Yeah, I didn't. Never mind. Pretend like I didn't do anything. So the stratum granulosum is at the boundary between the metabolically active cells below it and the layers below it and the dead cell layers above it. Let's go ahead and get that visualized. Okay, so we talked about the stratum basale, stratum spinosum, and now we're talking about this layer, the stratum granulosum. And if we look carefully, we do in fact see these little granules in there. And this is the boundary layer. These cells below this layer are still metabolically active. They're building things. They're involved in a bunch of metabolic activities. This layer of cell is beginning to die. These cell layers up here are dead. So that's the boundary between living tissue and dead tissue up here. And you can see, as I said, the laminar granules there in that layer. So this is and keratin is being organized within this region too. Okay, so let's come back to our drawing. And we'll establish this as orange. Okay. And that is the stratum granulosum. And the next layer is the stratum lucidum. That would be number 4. And this layer is present only in thick skin. And this layer is 4 to 6 cells thick. These are flattened, dead, and clear. And they contain, or it contains, large amounts of keratin and thick membranes, which tends to make it clear in appearance. And there's the stratum lucidum, this layer right there. So coming back to our drawing, you know what we're going to do? We're just going to leave it clear because it is the stratum lucidum. So let's go ahead and just label it. And maybe we'll just put in little dark lines here to indicate that layer. There you go. Okay. And now we come to the very last layer, the most superficial layer. And we're going to talk about that layer. And that's the stratum corneum. Number 5. The stratum corneum. Okay. And this layer is composed of around 25 to 30 cells on average, but it can vary. It can be from as few as or from a few cells, just a few cells, to 50 cells or more. So there's variation. The average is 25 to 30, but it can be as few as just a few cells thick. I don't know what they mean by a few cells, handful, 10, 15, whatever, and then 50 cells or more. So lots of variation. These cells are flat and dead. Okay. They contain keratin. Okay. They overlap one another. In other words, they have this overlapping structure. And the membranes of neighboring cells fit together like jigsaw puzzles. This layer is shed and replaced constantly. So this idea of these neighboring cells, so let me go ahead and see if I can get that drawn properly. I'm going to expand. I'm going to have one very large cell, and what you see is this kind of arrangement where one cell, its membrane is kind of like a puzzle piece with its neighbor, and we get this that kind of thing going on. So we get these cells that are fit together like puzzle. That kind of arrangement. They're not flat. They're not like pieces of wood just sitting flat against one another, but they have this wrinkly membrane that interlocks with one another. And it's a stronger connection between the cells when that happens. So let's go ahead, and so that's our last layer right there. These are dead keratinocytes, and that's the stratum corneum. Let's go ahead and fill that in. Stratum corneum. Which is our last layer. And what color do you think we ought to do? I don't know. Let's go with blue. There we are. That's the last layer. Okay. So that's it. Those are the 5 layers that make up the epidermis. I have one last piece of information that I would like to talk about that is related to the epidermis. And then our next lecture, because I'm going to break it off here, our next lecture will be on the dermis, and we'll finish up with the hair and the glands of the skin. But let me just finish this last little bit of information related to the epidermis. And what I want to talk about is keratinization. And basically we've already been talking about that as we've described the 5 layers and what is happening in each of these layers. That in totality is referred to as keratinization. So this is the process of the accumulation of keratin I'm getting tired still. Here we go. Okay. And the whole process taking place as cells move upward. Okay. So keratinization is these 2 ideas: the process of the accumulation of keratin and the process taking place as cells move up the layers, various layers. So this whole thing can be characterized as keratinization. Okay. The whole process of moving from the stratum basale and then sloughing off Takes about 4 to 6 weeks. Roughly speaking, then your skin it renews itself about every 4 to 6 weeks. And this is average. So some areas of the skin may be even quicker than this, depending on what kind of wear and tear it's getting. And sometimes in some areas may be less than that. Okay. So this replacement process is regulated through processes, or through mechanisms, I should say, that are not well understood. So how exactly the skin is replacing itself, at what speed it does so, and all of the triggers that are involved in this process is not completely understood. We do know that there is a factor called the epidermal factor, actually epidermal growth factor, that's involved, and this is abbreviated thusly. Okay, this plays a role. The main source of this, as a matter of fact, is the kidneys. So the main source of epidermal growth factor is this kidney, which is kind of interesting. Now, keratin, that protein we've been talking about, keratin comes in two forms. Number one, soft keratin. And this is more elastic. And that's the type that's found in the skin. The second type is what we would call hard keratin. And this is found making up our nails and our hair. So keratin is not just a single type of protein. We have variations. Soft, more like what we find in the skin, and very hard stuff, which our fingernails are made out of. The horn of a rhinoceros is hard keratin. Very tough material. Okay. All right, so the next lecture is going to start off talking about the dermis. And we will talk about that, describe its structure, and then we'll move into the functions. Again, as I promised, we're going to look at the functions in more detail. And then we'll look at the structure of hair in the skin. And we'll also talk about the glands of the skin. So that probably is going to be the next lecture. I don't think we're going to have 3 lectures here, as I said. So one more lecture, and I will see you there.